When muscles work out, they help neurons grow. Biochemical and physical effects of exercise could help heal nerves. MIT scientists find that motor neuron growth increased significantly over 5 days in response to biochemical and mechanical signals related to exercise. The green ball represents cluster of neurons that grow outward in long tails or axons. Credit, Angel. There's no doubt that exercise does a body good. Regular activity not only strengthens muscles, but can bolster our bones, blood vessels, and immune system. Now, MIT engineers have found that exercise can also have benefits at the level of individual neurons. They observed that when muscles contract during exercise, they release a soup of biochemical signals called myokines. In the presence of these muscle-generated signals, neurons grew 4 times further compared to neurons that were not exposed to myokines. These cellular-level experiments suggest that exercise can have a significant biochemical effect on nerve growth. Surprisingly, the researchers also found that neurons respond not only to the biochemical signals of exercise, but also to its physical impacts. The team observed that when neurons are repeatedly pulled back and forth, similarly to how muscles contract and expand during exercise, the neurons grow just as much as when they are exposed to a muscle's myokines. While previous studies have indicated a potential biochemical link between muscle activity and nerve growth, this study is the first to show that physical effects can be just as important. The researchers say the results, which are published in the journal Advanced Healthcare Materials, shed light on the connection between muscles and nerves during exercise and could inform exercise-related therapies for repairing damaged and deteriorating nerves. Now that we know this muscle-nerve crosstalk exists, it can be useful for treating things like nerve injury, where communication between nerve and muscle is cut off, says Ritu Raman, the Eugene Bell Career Development Assistant Professor of Mechanical Engineering at MIT. Maybe if we stimulate the muscle, we could encourage the nerve to heal and restore mobility to those who have lost it due to traumatic injury or neurodegenerative diseases. Raman is the senior author of the new study, which includes Angel Bu, Firdos Afgah, Nicholas Castro, Mahira Bawa, Sonika Kohli, Karina Shah, and Brandon Rios of MIT's Department of Mechanical Engineering, and Vincent Buddy of MIT's Koch Institute for Integrative Cancer Research. Muscle Talk. In 2023, Raymond and her colleagues reported that they could restore mobility in mice that had experienced a traumatic muscle injury by first implanting muscle tissue at the site of injury, then exercising the new tissue by stimulating it repeatedly with light. Over time, they found that the exercise graphed mice to regain their motor function, reaching activity levels comparable to those of healthy mice. When the researchers analyzed the graft itself, it appeared that regular exercise stimulated the grafted muscle to produce certain biochemical signals that are known to promote nerve and blood vessel growth. That was interesting because we always think that nerves control muscle, but we don't think of muscles talking back to nerves, Raman says. So, we started to think stimulating muscle was encouraging nerve growth. And people replied that maybe that's the case. But there's hundreds other cell types in an animal, and it's really hard to prove that the nerve is growing more because of the muscle, rather than the immune system or something else playing a role. In their new study, the team set out to determine whether exercising muscles has any direct effect on how nerves grow, by focusing solely on muscle and nerve tissue. The researchers grew mouse muscle cells into long fibers that then fused to form a small sheet of mature muscle tissue about the size of a quarter. The team genetically modified the muscle contract in response to light. With this modification, the team could flash a light repeatedly, causing the muscle to squeeze in response, in a way that mimicked the act of exercise. MIT scientists find that motor neuron growth increased significantly over 5 days in response to biochemical and mechanical signals related to exercise. The green ball represents cluster of neurons that grow outward in long tails or axons. Credit, Angel Bu. Raymond previously developed a novel gel mat on which to grow and exercise muscle tissue. The gel's properties are such that it can support muscle tissue and prevent it from peeling away as the researchers stimulated the muscle to exercise. The team then collected samples of the surrounding solution in which the muscle tissue was exercised, thinking that the solution should hold myokines, including growth factors, RNA, and a mix of other proteins. I would think of myokines as a biochemical soup of things that muscles secrete, some of which could be good for nerves and others that might have nothing to do with nerves, Raymond says. Muscles are pretty much always secreting myokines, but when you exercise them, they make more. Exercise is medicine. The team transferred the myokine solution to a separate dish containing motor neurons, nerves found in the spinal cord that control muscles involved in voluntary movement. The researchers grew the neurons from stem cells derived from mice. As with the muscle tissue, the neurons were grown on a similar gel mat. After the neurons were exposed to the myokine mixture, the team observed that they quickly began to grow, 4 times faster than neurons that did not receive the biochemical solution. They grow much farther and faster, and the effect is pretty immediate, Raman notes. For a closer look at how neurons changed in response to the exercise-induced myokines, the team ran a genetic analysis, extracting RNA from the neurons to see whether the myokines induced any change in the expression of certain neuronal genes. We saw that many of the genes upregulated in the exercise-stimulated neurons were not only related to neuron growth, but also neuron maturation, how well they talk to muscles and other nerves, and how mature the axons are, Raman says. Exercise seems to impact not just neuron growth, but also how mature and well-functioning they are. The results suggest that biochemical effects of exercise can promote neuron growth. Then the group wondered, could exercise's purely physical impacts have a similar benefit? Neurons are physically attached to muscles, so they are also stretching and moving with the muscle, Raman says. We also wanted to see, even in the absence of biochemical cues from muscle, could we stretch the neurons back and forth, mimicking the mechanical forces? And could that have an impact on growth as well? To answer this, the researchers grew a different set of motor neurons on a gel mat that they embedded with tiny magnets. They then used an external magnet to jiggle the mat and the neurons back and forth. In this way, they exercised the neurons for 30 minutes a day. To their surprise, they found that this mechanical exercise stimulated the neurons to grow just as much as the myokine-induced neurons, growing significantly farther than neurons that receive no form of exercise. That's a good sign because it tells us both biochemical and physical effects of exercise are equally important, Raymond says. Now that the group has shown that exercising muscle can promote nerve growth at the cellular level, they plan to study how targeted muscle stimulation can be used to grow and heal damaged nerves, and restore mobility for people who are living with a neurodegenerative disease, such as ALS. This is just our first step toward understanding and controlling exercise as medicine, Raman says.